Rightio. So basically, I've called this "Hooked on You," which I thought was quite an appropriate one for this. And what we're going to talk about is not just your traditional hookworms that everyone's heard about, but what's happening now and how we're starting to understand that these are really a lot of them actually zoonoses and require a One Health approach to control. So back when I was at university, there were two hookworms that infected humans: Ancylostoma duodenale and Necator americanus. I remember the first year out of uni, I was working as a relief scientist for PATH West, the West Australian State Health Labs. And I was up north, and one of the local scientists said to me, "If you want to control hookworm in Australia, you need to shoot all the dogs." And I didn't say anything, but being a young, arrogant new graduate, I thought, well, I know more than you because the hookworms of humans are only human specific and they do not infect other hosts. And I thought he was a silly old man for saying that. But in fact, he was absolutely correct, and I was the young, arrogant idiot because he obviously knew something that we didn't know at that time. Because really, you do need to, if you want to control the hookworms, you need to control the hookworm in the dogs. So as I said, traditionally there was Ancylostoma duodenale and Necator americanus. They were the two human hookworms. That's what I got taught at university, and that was where it ended. But things are a little bit more complicated than that. There are these hookworms of dogs. There are other hookworms in cats, but these are the hookworms that infect dogs: Ancylostoma ceylanicum, Ancylostoma caninum, and Ancylostoma brasiliensis. Also, Uncineria stenocephala can infect dogs in colder climates, but I'm not going to talk about them today. Now, these in dogs can cause different sort of things, but they all have a degree of public health importance. And I'm going to go through each of them and explain how they are relevant to human health. So the first and most important by far is Ancylostoma ceylanicum. So this was known to infect humans about a century ago, but it was considered to be this sort of rare, unusual zoonosis. Like so many of these parasites that we used to say were rare and exotic, and we're now discovering maybe aren't so much. It's a mean-looking thing. It's got these very large outer teeth. There's actually a very small two inner teeth, which you can't see in the plane of focus there. And back in the day when we used to identify these, you had to actually purge people who were infected, sieve their feces, find the adult worms that were passed out, and then get the microscope on just the right angle to look at the buccal cavity. So really, we didn't do that very often. It was a pretty complex process, and there were a lot of people who had hookworm back in the early part of the 20th century. So the actual speciation of hookworms in humans was very rarely performed. Now, in the new days of molecular sequencing, we can now do PCRs directly on your stool. So we're able to easily speciate hookworms from human and animal infections. And this is really changing our frontier and understanding of hookworm in humans. So the most important point is that Ancylostoma ceylanicum is a patent hookworm of humans. It causes patent infection, meaning that they sexually reproduce and the human passes eggs. This is a true human hookworm infection. It causes real morbidity. It causes significant malnutrition, particularly iron deficiency anemia. It's somewhere halfway between Necator and Ancylostoma duodenale, but sometimes healthy people can get quite significant iron deficiency with this hookworm. And look, if you're not actually doing PCR or some other way of speciating the hookworms, you won't know this is there. This is the second most common human hookworm globally, in my opinion. Ancylostoma duodenale is becoming quite rare in a lot of parts of the world. And in the Asia Pacific region, this is the second most common human hookworm. And it affects about 20% there, or even sometimes more of the human hookworm infections are this zoonotic strain. And how do you get it? Well, you can get it from cats and dogs, primarily dogs, but cats do carry it as well. And so now we have a problem because our traditional approach to soil-transmitted helminth control globally is we treat all the humans and we wait for the hookworms to go away. And I'm afraid that's not going to work. We're going to have to do a little bit more than that. Here's just a sort of brief timeline of what happened. It was ceylanicum in humans was first described by Lane in 1913. So we've known about this for over a century. But in the ensuing 50 to 60 years, it was perceived as a rare human infection, just occasional case reports, really not very important. There were volunteer studies and a number of case reports in the 60s through to the 80s, which suggested that this might actually be more common than we thought. But it was only really with the advent of the capacity to do molecular diagnostics and to easily speciate hookworms in humans that we started to realize its significance. I should have really done this as a circle where we have dogs and cats passing eggs and larvated hookworms hatching from them. And of course, these can infect through the skin. They live in contaminated soil, and under the right conditions, those infected larvae can live for up to 2 months in the soil. When you wander around barefoot, you're going out wandering around as we do barefoot. How many people walk barefoot in their backyard? I certainly do. Now, I don't have a dog. If I did, it might have Ancylostoma and I might get hookworm from it, or any other of these zoonotic hookworms I'm going to talk about shortly. And then it goes through the normal cycle of entering the skin, passing through the pulmonary or the cardiopulmonary circulation. You cough them up, they get in your guts, and then you end up with male and female hookworms mating and producing eggs. So this is a pretty standard hookworm life cycle, and the eggs are indistinguishable morphologically. So that's what a hookworm egg looks like. You see that, and you say, well, this person has hookworm. And the majority of laboratories will simply report hookworm. But we do need to think, if we're talking about public health control, what species of hookworm is this? This is tremendously important and has both clinical, but more importantly, public health implications. Let's talk a bit more about those clinical implications. Here is a picture of a Dutch marine in New Guinea in 1962. At that time, the western portion of New Guinea was still a Dutch colony, and there was a conflict going on between Indonesia and the Dutch. And eventually, of course, Indonesia did take over that region, and it's now part of Indonesia. But here's a picture of a Dutch marine. He's actually carrying an Indonesian rifle, an AK-47, which was not what they were issued. But here's the interesting thing. They had 11 hookworm-positive marines come back to Holland. 9 of them were found to be infected with Ancylostoma ceylanicum. And a number of these, 3 of them, were frankly anemic. They had frank iron deficiency anemia. And this is important because we're talking young, healthy marines. These people should be really, really fit and healthy, and they've still got iron deficiency anemia as a consequence of their hookworm. So, with something like Necator, you need to have a bit of an underlying deficiency to actually end up with IDA. But with Ancylostoma duodenale, you will become frankly iron deficient. And this is obviously the case also with Ancylostoma ceylanicum, because these young men had no other reason but for their hookworm infection to be iron deficient. So there were also experimental infections performed in the 80s, which showed that Ancylostoma ceylanicum can develop patent infection from percutaneous exposure. So it can get through your skin and cause a patent infection in your gut. And these people were all sick. They had abdominal symptoms, epigastric pain, flatulence. All of them had eosinophilia, as did the Dutch Marines. And so we know that this is a true human hookworm infection which causes significant morbidity. As I said earlier, we did a lot of things changed when molecular came out, and a man called Tawin Inkapau did his PhD on this in the mid-2010s under the supervision of Rebecca Traub, who's now at Murdoch University and is really the person who did a lot of the work proving that Ancylostoma ceylanicum is a human infection and a significant one. And they found that in some parts of Thailand, over 57% of the population had hookworm, and about half of these infections were Ancylostoma ceylanicum. In those regions, 90% of the dogs had Ancylostoma ceylanicum. They did do COX-1 sequencing and found these were the same sequences. So it really did confirm that this is a zoonosis from dogs in those regions. Here's a chart. It's updated from a review performed by Rebecca back in about 2021, which shows all of the surveillance that's been performed on humans for Ancylostoma ceylanicum globally since these reports in the early 2010s. And you can see that 50% of cases of hookworm, so this is a percentage of hookworm infections that were Ancylostoma ceylanicum, 50% in Cambodia and Vietnam, 36% in Ecuador, and we're going to talk about that shortly because traditionally we thought of ceylanicum as a Southeast Asian Pacific hookworm, but it's not. It's a global hookworm. Australia, 18% in one study, though that was a very limited number of actual infections. We only looked at about 11 infections there, so we only found 2 which were aceylanicum, one of which may have been important, but the other was almost certainly autochthonous. But it gives you the idea that this is certainly found around the world. And as I said, it was traditionally thought of as an Asia-Pacific problem, but there's been recent reports in humans from Ecuador, which is confirmed, and certainly it's present in Latin America. There have been reports from coyotes in Costa Rica, which suggests that there may be also a sylvatic cycle, and it's certainly present in dingoes here in Australia. And 41% of dogs in Grenada in the Caribbean were infected with this as well. One of the things I want to point out is that just because it's not meant to be somewhere doesn't mean it's not there. If you haven't looked, you won't find, and the absence of evidence is not evidence of absence. So it's certainly present in Latin America and in the Asia Pacific region, and it infects both dogs and humans there. It certainly infects dogs in the Caribbean region. Now, there have been these isolated reports from Africa, human cases in Madagascar and in Réunion, which is a French island close to Mauritius. And then we've had these funny animal case reports from Africa. Literally this morning, I saw an abstract appeared in Frontiers in Parasitology. The full paper is not yet available, from some work Adrian Street's group have done in Ghana, where they appear to have found human infections with Ancylostoma ceylanicum. So I think we can also now probably tick Africa off the list as somewhere where aceylanicum is endemic. And I'm coming to the conclusion that this is a global hookworm, and in many parts of the world, it's the second most common hookworm of humans. What's happening in our specific region? Well, Rick Spear invited me to do a trip with him to the Solomon Islands. We had a look at the hookworms there, and Rebecca Trout very kindly did the PCRs on them. And this found that aceylanicum is the second most common hookworms in the Solomon Islands in humans. Subsequently, Vito Collella's lab at University of Melbourne looked at dogs in Kiribati and found that it was very common in dogs in Kiribati. Now, there is unpublished data which will come out soon demonstrating that this is also very common in Papua New Guinea in humans. So we know that this is in the Pacific, and there is an expanding understanding that aceylanicum is very widespread in the Pacific, which means if you want to control human hookworm in the Pacific, you have to control hookworm in the dogs, and maybe the cats also. In Australia, Felicity Smouth did surveys in dogs and also in dingoes around the Cairns and Townsville region, and found a lot of aceylanicum in dingoes around Cairns, and I think also found in dogs in that region, certainly in dingoes. And they may be a sylvatic reservoir of aceylanicum in North Queensland. There are historical cases. We had these soldiers, Australian soldiers in the Northern Territory. There was a report from about 1944 of a number of Australian soldiers infected with Ancylostoma brasiliensis, which was synonymized with aceylanicum at the time. So they really probably mean Ancylostoma ceylanicum. It's just they had the same name back then. And all of these soldiers had been in Papua New Guinea, so we don't know if they acquired it there or if they acquired it in the Northern Territory. The only known likely autochthonous human infection with Ancylostoma ceylanicum in Australia was some work I did with Robin Gass's group. Hanson Kohler's the lead author on that, where we got a number of hookworm infections from Western Australia and genotyped them. This was in 2013, in the early days of this. And we found one human infection from the Pilbara region. Now, it's not published in the paper, but this was a very remote community in the Pilbara. It's very unlikely that that person had travelled. And so it's likely that that was an autochthonous case, but we were not able to follow up with the patient due to the limitations of the ethics. So certainly it's here in Australia. We don't seem to be having a lot of human infections. That's a bit of a mystery given that it's so common in dingoes. Maybe we're not looking and people are not being found with that infection, or maybe there's something different about our local ones that we're not as good at infecting humans. We really don't understand the transmission and the inferences of different genotypes, etc., on zoonotic transmission with this particular hookworm. The big issue is that somebody has hookworm, we say they've got hookworm, and we treat them, and we don't consider where they got it from, whether it's Necator, duodenale, or ceylanicum in human intestinal hookworm infection. So I think that qPCR identification or other methods of species-specific identification are going to be imperative as we move forward in hookworm control programs so that we can understand what we need to do. And one of the mistakes made in this was the very, very well-funded Bill and Melinda Gates Foundation D-worm-3 study in Malawi, India, and Benin, where they did PCRs for Ancylostoma duodenale and Necator americanus, but did not include a target for Ancylostoma ceylanicum. And we know ceylanicum is very common in India. We don't know what would have happened with those African samples. So perhaps a missed opportunity there to have a look at the implications of Ancylostoma ceylanicum in a large-scale study which was designed to determine the validity of mass drug administration for hookworm control. There's a very recently published paper, or published in the last 2 years, in Frontiers in Medicine, and this had a look at the effects of treating humans only as opposed to treating humans and dogs in an Ancylostoma ceylanicum endemic area. And you can see on the left, this is the prevalence of hookworm when humans only were treated, and the top line A shows when you do annual deworming, the bottom line C shows when you do biannual deworming. And on the right, you see what happens if you treat the humans and dogs. So there's a very stark contrast in how much you control hookworm depending on whether you're treating just the humans or the humans and the dogs if you're in an Ancylostoma ceylanicum endemic region. So we need to have a One Health approach to hookworm control anywhere where Ancylostoma ceylanicum is found in dogs or humans. Let's talk about some of the other ones. That's probably the most important, but I think we do ignore the other dog hookworms and their significance for human health. Ancylostoma caninum, beautiful looking thing here. It's got 6 teeth in its buccal cavity. Looks quite mean. There's one behind me here as my Zoom background. This used to be associated with cutaneous larvae migrans, but I personally think that's a bit of I don't actually think this really does cause cutaneous larvae migrans, which is when you have hookworm larvae crawling under your skin. The original studies, and I'll talk about this more in a moment, are not very reliable in their species identification. Caninum is a truly global dog hookworm. It's found in tropical, subtropical, and even some temperate regions globally. It's the most common dog hookworm globally as well. It does not cause patent infection in humans as far as we are aware. There are a small number of papers which talk about finding caninum infection in when they've PCR'd feces of humans who are passing hookworm eggs. There's some from India and some from South Africa. I suggest that those may be mispriming of the PCR on Ancylostoma duodenale. Ancylostoma caninum and duodenale are incredibly similar genetically, to the point where I suspect they should be subspecies of the same worm. And so you tend to find you get a lot of cross-priming in PCRs. So unless you've really sequenced the right region to differentiate them, I don't believe yet these reports of human egg passage of Ancylostoma caninum. But it remains a little bit of a question mark. We may yet come across true patent infection in humans. At this stage, I'm not convinced. However, it does infect the human intestine. It doesn't tend to reach patency, but you do get subadult forms, which can cause some significant issues for people. They cause something called eosinophilic enteritis, which is marked by abdominal pain and eosinophilia, and it can be quite long-lasting. Also, at the regions where they attach, they'll cause ulceration in the intestine. And I see Alex Lucas is in the audience, and Alex did a lot of work on this back in the 1990s, which helped define this particular, with John Cruz and some others, helped define this, Paul Prof, define this particular condition of eosinophilic enteritis caused by Ancylostoma caninum dog hookworms. The problem we have is these are not passing eggs. So the only way to really find it is to do colonoscopy on someone and find the worms in their colon or their jejunum. The second problem we have is, if anyone's had a colonoscopy done, is that you have PrEP, which means they give you a purgative and you have quite violent watery diarrhea for about 24 hours prior to your colonoscopy in order to wash out all of the fecal matter from your guts so the colonoscopy can be performed. It's strongly suggested in some papers that this also washes out the Ancylostoma caninum hookworms. So it's quite likely, in my opinion, that this is a very underdiagnosed condition, and that we might find, if we were actually able to somehow identify the presence of this in human feces, perhaps there will be highly sensitive molecular methods we could use to do that. We might find that it's far more common than we thought in those who have eosinophilia and abdominal pain. So the other interesting thing about this is Paul Prof and John Cruz and Alex started looking in Townsville in the early 2000s and the 2010s and thereabouts, I think back to the 1990s. And when they started looking specifically for Ancylostoma caninum in people who had symptoms which fitted the picture, they started to find a lot of these infections just in the Townsville region. And I'll show you some more about this. So if you'll pardon me for quoting the Bible in Matthew 7:7, it says, "Seek and you will find." And I think that's one of the take-home messages from this talk. Seek and you will find. You will find these things. The most recent report of Ancylostoma caninum infection in a human was one which I was involved with from Western Australia. This was a 35-year-old immunocompetent man from Perth. They had no significant travel history. They had been to Bali a little while earlier, but it was likely acquired in Perth. So during colonoscopy, they found aphthous erosions in the sigmoid colon, and they aspirated this live helminth. You can see the worm there on a standard microbiological slide. It's quite a small worm. And when you look at it up close, and it's quite difficult, by the way, to roll it around to get the right point on the buccal cavity, this is clearly an Ancylostoma caninum with the 6 teeth in the buccal cavity. And all hookworms can be speciated based on the morphology of the male bursa, which is the male sex organ that you can see on the right there. This was sent to Vito Colella's lab, where they performed COX-1 and ITS2 sequencing as well, just to really prove that this is what we thought it was. And it did come back as Ancylostoma caninum, as the morphology suggested. So there's the latest human infection. But I want to point out these are things which are occasionally picked up. Just like Ancylostoma ceylanicum 100 years ago, it was considered a rare and occasional human infection. I wonder if we might have a similar pathway in the coming years where Ancylostoma caninum eosinophilic enteritis becomes more and more commonly found and becomes recognized as a true common condition of humans. Maybe that'll happen, maybe it won't. In most cases, it's solitary or just a few subadult worms, but people do get quite nasty abdominal pain, etc., as a consequence of this. And if you see someone who's got that with eosinophils, you've got to start thinking caninum. Here are all of the reported cases in the literature where the infection was confirmed by recovery of worms. And you can see this goes right back to the 1920s. But look at the ones which I've highlighted in Townsville between 1989 and 1992. This is the time that John Cruz was looking actively for it. And over 1/3 of the human reports occurred during that short period of 3 years that John was looking for it. So if we look, we will find, and I'm sure there are many more cases of this globally than we realize. It's certainly exceptionally common with dogs globally. The final dog hookworm is Ancylostoma brasiliense. Now, it looks very similar to Ancylostoma ceylanicum, but the outer teeth are a bit shorter, and the buccal cavity is much longer than it is wide compared to ceylanicum. Because of the morphological similarities, Ancylostoma ceylanicum and Ancylostoma brasiliense were actually synonymized just as Ancylostoma brasiliense between 1925 and 1945. And then we realized, no, these are actually distinctly different species, and molecular sequencing has since proven that. I was working in Indonesia some years ago and talking to a local laboratory technician about hookworm, and he started talking to me about Ancylostoma brasiliense infections being common. And this was back in around 2010, before we'd really started to understand the prevalence of Aceylanicum. And I thought, that's a bit weird that you think that, but I didn't say anything. Well, the Indonesians still refer to this as Ancylostoma, or refer to Ancylostoma ceylanicum as a brasiliense in many cases in their textbooks. So it's likely that this is the Indonesians noticing the prevalence of ceylanicum in their region, but not necessarily providing the right name in some of the textbooks. Here's a map of these dog hookworms, and you can see that A. brasiliense, which is in red, is really quite restricted to the tropical and subtropical regions. This is a true tropical hookworm, whereas caninum is quite common in some temperate regions. See, it's been found in Melbourne and Perth and in Europe. And ceylanicum really does like warmer climates, but it can get a little bit cooler than A. brasiliense. A. brasiliense is really a tropical hookworm. And what does A. brasiliense do in humans? Well, unlike the other two, this one cannot get to the human gut. It enters into the host and it says, oh my God, I'm in the wrong host, and it doesn't know what to do. So the larvae just crawl around aimlessly under the skin. And they cause this condition called cutaneous larvae migrans. You can see some photos here I've taken in the Gambia on the left of an infant with this. It's not really got much of a reaction, but you can see the track of the larvae. In the middle is a friend of mine's child in the Gambia, and this child had a pretty significant reaction. We had one of our Peace Corps workers in a village down the road from me who had massive bullous, so blistering reaction to this. Some people get very severe reactions and are at risk of quite nasty secondary bacterial infections. And on the right is one of our JCU vet students who recently returned from Zanzibar and very kindly took photos of their cutaneous larvae migrans for myself and Con Constantino, our veterinary parasitology lecturer. So Braziliense is quite widespread in tropical regions in dogs and cats. It is found in certainly North Queensland and probably Christmas Island as well, but I suspect if we started sampling our dogs and cats carefully across the north, we'd find that it was present in most of tropical Australia. Any historical or foreign literature references to A. brasiliense causing pattern infection are likely that it's been either misidentified or synonymized, depending on the date, with Ancylostoma ceylanicum. But this shouldn't minimize the importance of cutaneous larvae migrans. This will go on for about 2 months. They will crawl under your skin. It's incredibly itchy. As I said, you're really at risk of secondary infection. And it's quite a nasty skin disease, which is quite difficult to treat because albendazole and mebendazole do not penetrate that well into the area of the tissue these things are. So you can give drugs, but it may not stop them crawling around. And often they just say, well, leave it, let it crawl around. One of my friends who's a local GP here says that he's had a lot of success. He sees this occasionally. He's had a lot of success using those freezing sprays that they use for warts and things, where he just goes to the advancing margin of the lesion, pulls it back about 10 centimeters and sprays the freeze on. Because remember, hookworms die below about 12 degrees Celsius. And he said he's managed to stop a number of the worms from crawling around in their tracks using that. Though that's not a officially verified and recognized treatment for it, seems to work for this GP. Here's a particularly nasty example of CLM there. You can understand why these people get secondary infections. So creeping eruption or cutaneous larvae migrans is probably uniquely a Ancylostoma brasiliense feature. It's the historical associations of caninum with creeping eruption, I think, are incorrect. And so I would suggest that only brasiliense causes true cutaneous larvae migrans like this. Here's just a summary slide talking about what these things do and where they're found. And if I give you one take-home message from this, it's that Ancylostoma ceylanicum is the second most common human hookworm certainly in the Asia Pacific, possibly in Latin America, and is probably found worldwide, infecting dogs, cats, and humans. So for hookworm control, we now need to consider a One Health approach, and we must speciate those hookworms using PCR or another method. Ancylostoma caninum causes eosinophilic enteritis and aphthous ulceration in the human gut and is likely very much underdiagnosed. And I think if we start looking, we will start finding a lot of cases. And finally, Ancylostoma brasiliense causes quite a significant dermatological condition and is somewhat under-recognized as a cause of disease. So hookworms in dogs and cats do matter, and they matter for human health as well. And finally, a One Health approach to hookworm control is going to be necessary in many parts of the world as we move forward. So that's my talk. We might leave questions to the very end. I'm now going to pass on to Xuan Zhao, who's going to talk more about hookworms from dogs and cats in Australia. And then we will pass on to Kate, who's going to talk about hookworms in dogs in Tonga. Thank you.